In this lecture, we will just continue with our discussion on the environmental requirements for seed germination. In particular, we will look at how temperature and light affect the germination of seeds. Another critical requirement for seed germination is temperature, which is a measure of the degree or intensity of heat as expressed in degrees Celsius or degrees Fahrenheit. The effects of temperature on seed germination can be defined in the context of cardinal temperature. What is cardinal temperature? Cardinal temperature refers to the range of temperatures that define the limits of germination. These temperatures are classified as cardinal minimum, cardinal optimum, and cardinal maximum. The cardinal minimum temperature is the lowest temperature that will allow germination. This temperature is difficult to determine because germination may actually be happening but at a very slow rate. Now, the cardinal maximum temperature is the highest temperature that will still allow germination. At this temperature, proteins essential for germination may start getting denatured or degraded. Within the minimum and maximum cardinal temperature, we have the cardinal optimum temperature. The cardinal optimum temperature is the temperature at which growth germination proceeds with greatest rapidity. In cotton, the cardinal minimum temperature for most cultivars has been identified as 15 degrees Celsius. This is equivalent to 0 degrees in cotton. The cardinal maximum temperature is 36 to 42 degrees Celsius, and the cardinal optimum temperature is 28 to 30 degrees Celsius. The germination responses of seeds to temperature will depend on several factors, including the species, the variety, the growing region, of course, the quality of the seeds, and duration of time to harvest. In general, seeds of temperate crops require lower temperature to germinate compared to seeds of tropical crops, which require higher temperatures. Seeds of wild species will have lower germination temperatures compared to domesticated species, and that could be an adaptation response to limiting environments. The seed is a reproductive unit, and its sole purpose is to ensure the perpetuation of the species. Lastly, high-quality seeds will have the ability to germinate under a wider range of temperature compared to poor-quality seeds, and I think this is self-explanatory. Naturally, if you have fully filled seeds that reach harvest maturity, you can expect that they will have better germination than seeds that's poorly filled and has not fully developed in response to some environmental stresses. Some seeds require daily fluctuating temperatures for optimum germination. This requirement for fluctuating temperature is commonly observed in seeds of species that have not had a long history of domestication. What do you think is the adaptive advantage of alternating temperature requirement? So imagine a natural rangeland where you have a rich soil seed bank. When seeds are buried deep in the soil and there is vegetation covering the soil surface, seeds are able to perceive a constant temperature. The constant temperature tells the seeds exactly that they are buried deep in the soil, and that if they germinate, they will have to use a lot of energy to break through the soil surface. Additionally, that they would have to compete with other vegetation. The necessity for alternating temperature, therefore, is to ensure that most seeds germinate on or near the soil surface in the absence of vegetation. The most commonly cited effects of alternating temperature requirement on germinating seeds is first, it creates a shift in the inhibitor-promoter balance where the inhibitor is decreased during the low temperature cycle and the promoter is increased in the high temperature cycle. Second, it changes the structure of seed components, which in the original form prevents germination. Other species, particularly temperate plants, require stratification or pre-chilling. Stratification is a temperature preconditioning of imbibed seeds in cool, moist condition to promote germination. This graph shows the germination of loblolly pine after different days of stratification. We have here 0, 15, 30, 45, and 60 days of stratification. From these line graphs, we can see that germination in loblolly pine reached its maximum of around 85% between 13 and 15 days following stratification for 30 to 60 days. While some seeds require cold exposure to germinate, others, especially those of tropical plants, are susceptible to cold exposure. When germinating seeds of tropical plants are exposed to low but non-freezing temperature, they suffer from chilling injury. In cotton, for example, chilling injury results in seedling malformations, taproot loss, reduced vigor, increased occurrence of seedling diseases, poor seedling establishment, and ultimately to low yield. Let's look at the cellular basis of chilling injury during germination. For this, you will have to remember your cell structure from your biology class. This is a plant cell. Plant cells are enclosed in a membrane, which is of course called the cell membrane, and that is this structure, the thin green line that you see right here. The cell membrane is a complex structure composed of two layers of lipids that looks like the two slices of bread in this panel. So this one here and also this layer. Within the layer of lipids, you can see proteins and carbohydrates and complexes between the two. Now, this lipid bilayer that looks like a sandwich is the most universal organization of a cell membrane. This lipid organization is also known as the lamellar phase. Here's another illustration of a cell membrane where the lipids are arranged at a lamellar phase. In seeds, the membranes are not arranged like this. Instead, they are arranged like this. This lipid organization is known as the hexagonal phase. A lipid is composed of a polar head and a fatty acid tail. This is an example of a lipid. You have here the polar head and you have here the fatty acid tail. The membranes rearrange like this during seed maturation because it needs to get rid of excess moisture. If the membrane in a dry seed is arranged like this, it will not be able to get rid of excess water because these polar heads will hold on to water molecules. But in a hexagonal phase like this, the polar heads are directed towards each other, as you can see here. They surround whatever little moisture is left in the seed. Now, during water imbibition, the cell membrane in dry seeds rearranges itself from a hexagonal to a lamellar phase. Now, during this transition, at normal temperature, solutes are going to leak out of the cell. It's like turning a grocery bag inside out. If you have stuff inside the bag, it will fall off. So it's just like a membrane reorganizing from a hexagonal phase to a lamellar phase. There's going to be leakage of solutes out of the cell. Under cold conditions, the reorganization becomes slow because the membrane loses flexibility. When the membranes become rigid, it will be difficult to incorporate new lipids to support the expansion of the membrane. This results in lesions or breaks in the membrane, which in turn exacerbates or worsens leaking of solutes out of the cell. In addition, the ruptured membranes disrupt the enzymatic conversion of stored reserves to energy. All this results in poor germination. From temperature, let's move on to light. From our previous lessons, you already know that light has several properties, including duration or length, intensity, and quality. Day length refers to the recurring cycle of light and dark periods within a 24-hour cycle. Light intensity refers to the total amount of light that the plants are exposed to. This light property can be measured in micromoles per square meter per second. Then we have light quality. Light quality refers to the spectral composition of light in terms of the wavelengths that are critical in plant biological processes. Specifically, these are the wavelengths within the 400 to 700 nanometers of the light spectrum. These wavelengths are important for photosynthesis, and that is why this region of the light spectrum is called photosynthetically active radiation, or PAR. Let's now see how seed germination is affected by these different properties of light, starting from day length. Some seeds exhibit photoperiodically controlled germination response. This control of germination is activated by phytochromes that are similar to those activated during flowering. What are phytochromes? Phytochromes are multifunctional proteins that serve as the eyes of the seeds. The phytochromes can sense light, and they tell the seed if it's time to germinate based on the duration of exposure of seeds to the light. Seeds that require prolonged exposure to light to germinate are called long-day species. Seeds that do not require prolonged exposure to light to germinate are called short-day species. There are some species of plants that doesn't require light to germinate, and a good example of that is radish. This slide illustrates the effects of day length on the germination of 4 different rhododendron species. Rhododendrons are ornamental plant species that produce very showy flowers, as you can see in these panels. So in this example, we have Rhododendron breviperulatum, Rhododendron ovatum, Rhododendron keneheri, and Rhododendron simpsonii. The graphs show the germination rate of each rhododendron species after exposure to 0, 1, 4, or 16 hours of light. Germination was measured within a period of 30 days. Based on the results, we can say that all 4 rhododendron species require light to germinate. In the absence of light, none of the 4 rhododendron species germinated after 30 days, and that is indicated by the line graphs with the boxes here, 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 and also here. These lines graph the germination rate of rhododendron that were not exposed to light, and as you can see, they remained 0. In contrast, exposure to 16 hours of light induced the highest germination in rhododendron breviperulatum, ovatum, and simsii. That is indicated by the line graph with the circles. So that is this line here, here, and also we have it here. After 30 days, germination in these 3 rhododendron species reached more than 80%. Rhododendron kanehirae also germinated well after exposure to 16 hours of light, as indicated by the graph with the circles. It's the line right here. But its germination was even better when exposed to only an hour of light, as indicated by the line with the diamond. So that is this line over here. Now, these variable responses of the different species of rhododendron is controlled by the genetic makeup of each species. This slide illustrates the effects of light intensity on the germination of 4 different rhododendron species. Again, we have rhododendron breviperulatum, rhododendron ovatum, rhododendron kanehirae, and rhododendron simsii. The graphs show the germination rate of each rhododendron species after exposure to variable intensity of light, specifically to 0.1, 1400, 700, and 3200 lux of light. Lux is a unit of light intensity or illuminance. So again, germination was measured within a period of 30 days. Based on the results, we can say that all 4 rhododendron species require light to germinate. In the absence of light, none of the 4 rhododendron species germinated after 30 days, and this is indicated by the line graph with the boxes. So that is that line here, 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 and also here. So you can see the germination stayed at 0. When exposed to 700 to 3200 lux, all seeds germinated, although at varying rates. At the end of the 30-day period, we can see that R. breviperulatum, ovatum, and simsii had more than 80% germination. In contrast, rhododendron kanehirae has around 70% germination. Overall, this data shows us that different species will have different germination responses to light. Lastly, we have light quality. This slide illustrates the effects of red, blue, white, and a combination of red and white light on the germination of Stevia rebaudiana, and that is the plant that is pictured right here. On the y-axis of these graphs, we have the germination rate, and then on the x-axis, we have the number of days of germination. Seeds of Stevia were germinated in moistened filter paper or agar at 20 or 25 degrees Celsius. Graph A shows the germination of Stevia seeds in filter paper substrate at 20 degrees Celsius. Graph B shows the germination of Stevia seeds in filter paper at 25 degrees Celsius. Graph C shows the germination of Stevia seeds grown in agar at 20 degrees Celsius, and Graph D shows the germination of Stevia seeds in agar at 25 degrees Celsius. So in general, we can say that Stevia seeds responds differently to the treatment combinations as illustrated by several lines in each of these graphs. But when grown in moist filter paper at 20 degrees Celsius, we can see that germination is highest when the seeds were exposed to blue light, and this is illustrated by this line right here with the boxes. But under 25 degrees Celsius, we can see that germination was better under white light, as indicated by this line here with the triangles. When grown in agar at 20 or 25 degrees Celsius, germination was best when seeds were exposed to blue light, as illustrated by the line here with the boxes, and also here, again with the boxes. From this slide, we can see how the combination of light quality, temperature, and even substrate induce variable germination responses from Stevia. In summary, seed germination is affected by the genetics and physiology of the seed as well as the environment. These environment factors include water, air, temperature, and light. The germination responses of seeds to the environment are closely related to the evolutionary adaptation of the species. So if you have a tropical plant that is adapted to relatively warmer temperatures, you can expect that the seeds of this plant will also require warmer temperature and maybe even higher humidity to germinate compared to seeds of temperate crops.